Hello my dear students, welcome to the MS Science Academy. Myself Minu Srivastav, and today we are going to discuss about the active transport. In the previous lecture, we have discussed about the passive transport. So in this lecture, we are going to study about the active transport. As the name suggests, the active transport, active means the substance or the molecule we have to activate, and any molecule or any substance, if we want to activate that particular substance, we have to give the energy. So this, in the active transport, energy is required. Okay, so energy is required. Okay, so in this process the energy is required, and this process is known as the uphill process also. As we have seen in the passive transport, the passive transport is known as the downhill process, whereas the active transport is known as the uphill transport. This is the uphill transport. Okay, so this is the uphill transport. So active transport, these are the transport system which require the energy, and it is known as the uphill transport also. This kind of the definition you will write. Okay, this is the transport system which require energy, and particular transporter proteins are required in this process. Means in the active transport, particular transporter proteins which are embedded already in the plasma membrane. So these proteins help to transport in a particular way from either the outside or the inside the membrane. Okay, so particular transporter proteins are required. So this is a transport system which require energy, and transporter proteins help in this process. Okay, so here the transporter, okay, this is all for the active transport definition. But the active transport system is of two types: primary active transport and the secondary active transport. The primary active transport is differ in the primary and the active transport, both are required the energy, but how we can utilize the energy, this type of the main difference is this, that how we can utilize the energy. Okay, so active transport, I am writing AT. Active transport is of two types. This is the primary active transport and secondary active transport. In the primary active transport, particular energy is required by the energy which is required, which are coming by the hydrolysis of the ATP. Okay, so energy by hydrolysis of ATP means ATP, adenosine triphosphate. Okay, so ATP is the energy which is required. In the primary active transport, the ATP is hydrolyzed in the form of ADP, and this way energy is produced. Okay, whereas in the secondary active transport, the energy is stored, and by this energy, particular ionic concentration gradient will be reformed, and different antiporter and the symporter molecules help to move the molecule either the inside or the outside. Okay, so here we will write the energy by stored form in an ionic concentration gradient. And as the ionic concentration gradient will be generated, particular symporters and antiporters are helped to move the particles. Okay, so in the secondary active transport, we will discuss about the symporters and the active transport, symporters and the antiporters. Okay, now we will discuss about the primary active transport. Okay, so in the primary active transport, we will discuss about the sodium potassium pump. Now we will discuss about the primary active transport. As we have discussed that the primary active transport utilize the energy by the hydrolysis of ATP. Okay, so here the energy is utilized by the hydrolysis of ATP. Okay, so in the primary active transport, the energy comes by the hydrolysis of ATP. Due to the energy, the shape of the transporter proteins, which are named as a pump also, which change their shape and help the transfer the material or the substance against the concentration gradient. Okay, so here we will write due to energy shape of transporter proteins or pumps change. Due to energy, shape of transfer proteins change, which help to transport the material against the concentration gradient. Okay, means the material transport against the concentration gradient. This thing, what is the concentration gradient, we have discussed in the previous lecture. Okay, so I am not going again with this topic. So here in the primary active transport, the materials is transport against the concentration gradient. So in the primary active transport, the best example is a sodium potassium pump. Example, that is sodium potassium pump. What is a sodium potassium pump, or we can say the sodium potassium transporter protein? The sodium potassium pump, if you have read something about the nerve impulses, in that nerve impulses, the 3 sodium outflux and 2 potassium ion on the behalf of 3 sodium ion, 2 potassium ion influx into the membrane, and like that the nerve impulses carry forward. So here the transporter protein which help to 3 sodium and the 2 potassium deals, this is known as the sodium potassium pump. Now we are going to discuss about the mechanism of sodium potassium pump. 3 sodium pump in such kind, I will describe with the help of this diagram. 3 sodium pump, 3 sodium ions bind with this sodium potassium pump. This sodium potassium pump has the activity of sodium potassium ATPase, and this sodium potassium ATPase just convert the ATP into ADP, and this phosphate molecule just bind with this. As the sodium potassium ATPase activate, this 3 sodium ion bind, and as the phosphate molecule attach, this shape of the transport protein or the pump molecule change. So this 3 sodium expelled out. As this 3 sodium expelled out, in this changed shape, 2 potassium ion attached, and the phosphate molecule just removed out, and as the phosphate molecule removed, 2 potassium ion expelled in. 2 potassium ion just introduced into the cytosol. Okay, so here 3 on the behalf of 3 sodium ion, 2 potassium ion inhale in, and the 3 sodium ion expelled out. Okay, so here 3 I will write the mechanism. 3 sodium ions pump, 3 sodium ion not pump, bind to pump, and during this ATP convert into ADP. Okay, so here ATP convert into the ATP, so leads the change the shape of pump and expelled out 3 sodium. Okay, here simultaneously 2 potassium ion bind to pump, 3 sodium and 2 potassium ion bind to pump and phosphate group removed. And again regain the shape of pump. So 2 potassium ion transfer into cytosol, okay, or cytoplasm. So like that, 3 sodium and 2 potassium ion transport. Now we will discuss about the secondary active transport. Now come to the secondary active transport. In the secondary active transport, as we have discussed in the secondary active transport, the stored form of the energy, we can utilize the energy which is already in the stored form in an ionic concentration gradient. What is the ionic concentration gradient and how we can maintain, we will discuss here. Okay, here the secondary active transport, the energy stored in the sodium and the hydrogen ion concentration gradient is used to drive the substance across the membrane against the concentration gradient. Here in the secondary active transport, again we have to go for the primary active transport, because in the primary active transport the sodium concentration is maintained outside the plasma membrane, or we can say the extracellular site. Here the sodium ion concentration is maintained with the help of pump, sodium potassium pump. So as the sodium ion concentration is higher on the outside, it will lead the secondary active transport, and the sodium concentration stored the energy in the form of potential energy, and this potential energy is converted into the kinetic energy. Ke represent the Ke kinetic energy, and these kinetic energy help to drive the substance or the ions move in or move out from the membrane. Here sodium potassium pump maintain. So here the sodium concentration, sodium ion or the hydrogen ion concentration is very much important to maintain the concentration gradient, and this sodium ion concentration gradient is maintained with the help of primary active transport, with the help of sodium potassium pump. So be careful, the secondary active transport will not proceed without any help of the primary active transport, because the secondary active transport is totally dependent on the primary active transport. So the help of the primary active transport maintain the sodium concentration, and the sodium potassium pump maintain the sodium concentration gradient across the membrane. The sodium ion concentration gradient is maintained across the membrane. So the sodium ion have the potential energy, and this potential energy is converted into the kinetic energy. Okay, so here this kinetic energy help to move the substance. Okay, here this kinetic energy leads the change in the shape. Okay, so a transporter protein bind to the sodium and the another substance. With the help of this potential energy, the change in the shape of pump proteins take place. So here the sodium ions and the another ion will bind with the that pump proteins, and either they will move same direction or they will move against the direction, means in opposite direction. With the help, if they are moving in the same direction, they are naming as the symporter. We are talking about whom? We are talking about the sodium ions and another substance, means the sodium ion must be present. Means if we are just taking H ion, if we are taking the calcium, if we are taking the another ion, amino acids or the glucose, other substance also, but for that movement sodium concentration must be important. Okay, so here symporter and antiporter, if we are discussing that move in the same direction, move in the opposite direction. So how many ions will move in the same direction? 2 ions. 2 ions means 2 respective ions, sodium and another substance. If we are saying that the antiporters move the ions in the opposite direction, which are the two ions? Sodium ion and the another one. Means what I want to say that the sodium ion concentration must be essential. Means whatever ion, the calcium ion, H ion, that glucose, amino acid, whatever, but with these molecules sodium ion must be represented. They will hold the finger of the sodium ion. Okay, so here the symphotos move in the same direction, okay. So here like sodium and glucose, sodium and amino acid. And if we are saying that antipotar move in opposite direction, example sodium, and if sodium is moving inside the plasma membrane or the cytosol, the calcium +2 ion move in the extracellular site. And if this is the one example, sodium and calcium, and the another example is sodium. If sodium is moving inside, and the H ion concentration will move in the outside the membrane. So here the transporter proteins bind to the sodium and another substance. This change the shape, and the when they are changing the shape, they can move and they can help to move with the two mechanisms, symphotor mechanism and the antipotar mechanism. In the symphotor mechanism, two ions move in the same direction, like the sodium and the glucose, sodium and the amino acid. And if they are moving in the opposite direction, sodium and the calcium +2, sodium and the H ion concentration. So I am representing with the diagram also. Here the plasma membrane, and here some transporter proteins I have shown. Here the sodium ion moves inside the cytosol, and the calcium ion move out, okay, and the sodium ion move in and the hydrogen ion move out. And here the sodium and glucose same in the direction, flow in the same direction, and the sodium and the amino acid in the same direction. So here in the secondary active transport, the concentration gradient, ionic concentration gradient, especially sodium ion and the H ion concentration gradient is must be essential. So with the help of sodium concentration gradient, the symphotos and with the help of symphotos and antipotar mechanism, we will move the ions and the material. This is whole for the primary active transport and the secondary active transport. Now we will discuss about the transport with the vesicles like exocytosis, endocytosis and all. Now we are going to discuss about the transport in the vesicles. The vesicle word represent the sac-like structure. It represent the sac-like structure. Here the transport in the vesicle is the utilize the energy, and the energy is utilized with the form of ATP. Okay, so transport in the vesicles is with the help of two mechanisms. One is the endocytosis and second is the exocytosis. One is the endocytosis and second is the exocytosis. As the word represent endo means inside the cell, inside, and cytosis means cell. Means the material is transported towards the membrane or inside the cell. Means the transport vesicles inside the cell. Transport the vesicle inside the cell, or and in this process the vesicles is detached from the plasma membrane. The vesicles formed by the detachment from the plasma membrane. Okay, so here in the endocytosis process, the material is transported in the vesicle, and these vesicles are formed by the detachment of the plasma membrane. Now come to the exocytosis. Exocytosis, the words represent the exo means exit and cytosis means the cell. So here the transport vesicles outside the cell, means the vesicles are transport from the outside. Okay, so here the vesicles are formed by the fusion with, या इसको आप with लिखिए, with जो with the fusion of cell membrane. It means the exocytosis process, here the vesicles are formed by the fusion of the cell membrane, and in the endocytosis process the vesicles are formed from the detachment of the cell membrane. Now we will discuss about in the very much detail about the exocytosis and the endocytosis. Endocytosis process has the three types. First is the receptor mediated, second is the phagocytosis, and third one, that is bulk phase. Okay, so this endocytosis are of the three type. One is the receptor mediated, second phagocytosis, and third bulk phase. So here we will discuss about the receptor mediated. As the name suggests, the receptors are mediated such kind of the endocytosis process. Here we will discuss about the LDL receptors, LDL mediated receptors. Here the LDL receptors, LDL means low density lipid, for example cholesterol. LDL receptor complexes are present on the plasma membrane, and these receptors are susceptible for the LDL. Okay, so LDL molecule will bind with this receptor, and these receptor and LDL form receptor LDL complex. This is the receptor LDL complex, and these receptor LDL complex will bind to a particular site. These are the clathrin coated pin. What is the clathrin? Clathrin is a particular protein, and this receptor LDL complex will bind only on the clathrin site. So when this receptor mediated complex bind on the clathrin coated pit, the cell membrane invaginate and form. This invagination membrane will attach or fuse and form a vesicle. This is known as the clathrin coated vesicle, and here just this complete endocytosis process happen complete with this clathrin coated vesicle. And after this, uncoating take place. Uncoating means the clathrin molecules, clathrin protein molecules will remove from this vesicle and approach towards the plasma membrane. And here the uncoating, as the uncoating happen, these uncoated vesicles, this is the uncoated vesicles, fuse with the endosome. Why this? And when the uncoated vesicle fuse with endosome, these here the LDL separate from the receptors, means after the fusion with endosome, the receptor molecules are separate from the cholesterol or the LDL molecules, and these receptors will again approach towards the membrane. And here only in the vesicle only the LDL molecules remain. After this, the lysosome will attach with these vesicles which have only the LDL molecules, and these lysosomes will break down the large lipid and the protein molecule. Here I have written all the steps. First is the binding. The cholesterol bind with the receptor, and receptor LDL complex will form. Here the vesicles formed called clathrin coated vesicles. Here particular vesicles are there, and which are the clathrin coated vesicles. Okay, now in the next step the uncoating happen. Here the clathrin molecules remove, and this vesicle is known as the uncoated vesicle. This is the uncoated vesicle. Here after the uncoating, the fusion with endosome take place. The uncoated vesicles and the endosome, this process help the LDL separate from the receptor. The LDL separate from the receptor. So here the LDL separate from the receptors, and these receptors again approach towards the plasma membrane. Again, here the recycling of the receptors, these receptors approach towards the plasma membrane, and after this in the vesicle, after this only the large protein and the lipid molecules who is attached is only present. So the lysosome will break down, lysosome will attach, and with the help of enzymatic process these molecules will break down. Okay, this is all for the receptor mediated endocytosis. We will discuss about the bulk phase and the phagocytosis. Now we are going to discuss about the endocytosis process. In that endocytosis, we will discuss about the phagocytosis. Phagocytosis means the cell engulf the solid part such as the bacteria, viruses. Generally, the macrophages and the neutrophil cells, such cells are showing such kind of the process. If in the lower classes, if I think you have seen the amoeba nutrition, as in the amoeba nutrition, amoeba engulf their food, amoeba engulf its food with the help of pseudopodia. Pseudopodia are just nothing, they are the finger-like projections of the cell membrane. Here, the particles bind with the plasma membrane receptors, means the particular receptors or proteins are present on the plasma membrane, and the particles or the bacteria, virus are when bacteria and virus just attaching with that receptors, such cells showing the pseudopodia formations, means such cells are showing the extension to engulf that particular material. Here this is the pseudopodia, here that is the pseudopodia, and this is the receptor, and here these are the like bacteria and virus, whatever material which we have to engulf. Okay, so here the particles bind with the plasma membrane receptors and the pseudopodia extension occur. Here after the pseudopodia engulf process, the phagosomes are formed. The phagosomes are formed. This is the phagosomes, means when the bacteria and virus engulf with the help of pseudopodia, these cell membrane just form a vesicle or sac-like structure. This is called the phagosome, in which the bacteria and virus or whatever material trapped out. Okay, here this with the help of this phagosome enter into the cytoplasm, and the lysosome will fusion with lysosome. Okay, here the lysosome will fuse, and the lysosomal enzyme digest this material, this bacteria, virus, whatever. Lysosomal enzyme break down the indigested material. Okay, here the digested material diffused in the cytoplasm, means the digested material diffused in the cytoplasm, and the undigested part remain in the vesicle, and that part is known as the residual body. Okay, so means the body means the phagosome, it's a kind of the phagosome, it's a phagosome which has the undigested part, means the residual body is just nothing, it's a phagosome which is having only the undigested part. Okay, so and that's this residual body is just throughout our cells with the help of exocytosis. Now we will discuss about the bulk phase endocytosis, that is the pinocytosis. Now come to the pinocytosis, or we can say the bulk phase endocytosis. It is the process in which the cell drink or cell take the liquid materials, especially here the liquid or the tiny droplets are taken from the extracellular fluid. So here I will write the definition: tiny droplets are taken up from extracellular fluid. Okay, so here tiny droplets are taken in the form of the vesicle. Here the main thing is that there is no receptor proteins are involved in this process. All the solute dissolved in the extracellular fluid brought up into the cell in the form of vesicle. Here the solid particles or the fluid is taken in the form of vesicle. After the formation of vesicle, the lysosome will, this vesicle detach from the plasma membrane. PM means it's not prime minister, it's plasma membrane, and it's enter into the cytosol. This vesicle will enter into the cytosol, and the lysosome will fuse. Lysosome will fuse with this vesicle, and here the digestion take place by the lysosomal enzyme, and the digested solids will diffuse throughout the cell. So in this process, the cell or tiny droplets, or we can say the small particles, very small particles are taken up, and it's also known as the cell drinking process also. Okay, so after this we will complete the exocytosis, and by this now come to the last part, that is the exocytosis. As the word represent, exo means exit and cytosis means cell, means the something or whatever material is omitted out or removed from the cell is known as the exocytosis. If you want to understand the exocytosis in the simple word, it's a kind of vomiting. Means the cell vomit out some material and release such kind of the neurotransmitters, mucus and other secretions, enzymes and whatever. So in the exocytosis, the cells releases such kind of the material by diffusing out by fusion of the vesicles with the cell membrane. Here the release of the material from a cell, there are generally two types of the cells like secretory cells and nerve cells showing such kind of the process, exocytosis. Here the secretory cells such as the enzyme, hormones, mucus, such materials, enzymes, hormones, mucus, such kind of the secretory cells are showing such exocytosis, and in the nerve cells neurotransmitters are released with exocytosis process. Here the membrane enclosed vesicles are known as the secretory vesicles formed inside the cells. Means such secretory vesicles are formed inside the cells, and these secretory vesicles fused with the membrane. Here I am showing one nerve cells. The nerve cell, or we can say the axon terminal, having the neurotransmitters, and these neurotransmitter releases from one nerve cell to the another cell with the help of exocytosis. Here the secretory cells which is having the neurotransmitters, these are having neurotransmitters, and here this neurotransmitters, secretory vesicles which are having the neurotransmitters fuse with the cell membrane or nerve membrane, and these nerve membrane they fuse with the nerve membrane and releases their neurotransmitters. So here the membrane enclosed vesicles known as the secretory vesicles formed inside the cell, fuse with PM means plasma membrane, and releases their content in the extracellular fluid. This is the area of the extracellular fluid, and these materials will attach with the another new membrane. Okay, so this kind of the exocytosis process happen in generally neurotransmitters with the help neurotransmitters and secretory cells like enzyme, hormones, mucus, like that. So this is overall transport of the across the cell membrane. In the next video we will see the cell division. Till then, thank you, bye bye, stay safe, stay healthy, thank you.